Hello everyone, thank you so much for coming to this video. I'm Nicholas J. Parody, and I collaborated with Dr. Chung Wu, Philip Lekernick, Maria Herb, and others in developing a new theory of molecular evolution to help describe the evolution of SARS-CoV-2 virus. This is called the near-neutral balance selection theory, and its implications on the neutralist-selectionist debate for the last 50 or so years. Now, of course, we have to delve into the pandemic, which is currently caused by severe acute respiratory syndrome coronavirus 2, also known as SARS-CoV-2. The COVID-19 pandemic began in December of 2019 after the emergence of SARS-CoV-2, which in itself is a single-stranded RNA virus of approximately 30,000 nucleotides, or bits of genomic information. You probably know this as adenine, thymine, cytosine, and guanine, and with RNA, uracil replaces thymine. Collections of nucleotides make up the coding genes and non-coding gene regions. What do I mean by this? This basically means that coding genes eventually translate into protein, which are made up of nucleotide triplets, or amino acids, that contain biological functionality. Conversely, non-coding gene regions, such as the untranslated region or translation regulatory sequences, do not translate into protein, but instead regulate the expression of coding gene proteins, or stabilize the genome. In this, SARS-CoV-2 contains about 10 coding genes, some which serve structural roles, such as S, E, M, and N, or spike, envelope, membrane, and nucleotide capsid proteins. There are others that mediate viral replication, open reading frame 1AB, which is composed of 15 non-structural proteins, or NSP 1 through 15. Also, some other proteins exist which are believed to alter the host immune response upon viral infection, that being ORF3A, ORF6, ORF7A, ORF8, and ORF10. Now, how exactly does the virus create these proteins to basically do their job? In replication, SARS-CoV-2 undergoes transcription within the host cell. It first has to infect the cell in order to start transcribing or replicating anything. During transcription, the RNA genome is copied from the template strand from the 5' end to the left to the 3' end to the right using an RNA-dependent RNA polymerase known as RDRP. This strand is proofread for any errors using a 5' to 3' exoribonuclease. RDRP uses the conserved TRS, transcription regulatory sequences, as signals in order to transcribe each gene segment. So when it reaches one of these TRS signals, it basically stops transcription, and then that gene will later then become translated. But basically, following transcription, translation by the host cell ribosome and post-translational modifications will then generate mature viruses, which will then bud out from the cell and infect other host cells. SARS-CoV-2 is known to infect many tissues. One of the most prominent places are the lungs. And already 3 years into this pandemic, the virus has caused over 6 million deaths and over 600 million infections worldwide. And recently in the news, we heard about the infection surge going on in China when they lifted their no COVID policy back in December. This, if not controlled well enough by China, as well as the international community, could spark another global infection wave, thus indicating that this pandemic is far from over. To get this pandemic under control, we must understand the molecular evolution of SARS-CoV-2. Genomic databases such as NextStrain are actively tracking the evolutionary progress of SARS-CoV-2 by creating a phylogenetic tree. The idea is that from the beginning of the pandemic, the first ancestral strain of SARS-CoV-2 in the leftmost region of this diagram, by that point at December, can mutate following replication, generating variations in the virus population. Now, some mutant strains are considered not viable and are eliminated from the population. However, those that do survive may contain favorable mutations, therefore living on and reproducing to perform more mutant variants. When the mutants become fixed in the population, a new phylogenetic clade arises and diverges from the ancestor, thus representing a branch in the tree. These clades could be more or less infectious, deadly, or hardier to the host environment than their ancestor. Because of this, more clades can emerge over time and become dominant in the virus population. If you can play this video, you'll notice that the emergence of a different virus clade globally becomes more apparent over time between December of 2019 and July of 2021, the date range in which we conducted our study. From this, we can see that the alpha, beta, gamma, delta, kappa, epsilon, eta, iota, and lambda clades each have their own branch, with at this point both beta and delta being the most dominant strains. While not explicitly labeled, the Omicron strain is becoming emergent, beginning all the way back in March 2021. However, it was not the dominant strain at the time. Now we know, of course, that in the media, the Omicron is pretty much the dominant strain globally. Okay, so we briefly talked about favorable and non-favorable mutations and how they're observed in the population. I must make a distinction here that when a mutation is observed in the population and is not removed, we call this a substitution. A mutation that is removed from the population is not a substitution. This will be important. Different mutations are either kept or removed based on how they impact fitness or an organism's ability to successfully reproduce. Harmful mutations can decrease fitness and be potentially deadly, like if you were born with some rare genetic disease that you were born without most of your organs, you won't be able to live long past the birthing state. They are thus removed by negative selection and are not observed. Neutral mutations do not impact fitness and have equal chance as non-mutants to be observed. Thus, they are either equally likely to be observed. Now, with foreshadowing, under neutral selection, the mutation rate is thus equal to the observed substitution rate. This will be important. Beneficial mutations increase fitness and are likely to be observed under positive selection. This type of selection is actually quite rare, but this is what allows evolution to occur. This allows change in an organism from its initial state. Let's go back to SARS-CoV-2 and note something interesting. We notice that there is a constant mutation rate for the SARS-CoV-2 genome, despite changing environmental factors, such as changing number of infected humans in the global population and the amount of vaccines that have been handed out since the beginning of January of 2021. Now, we noted that the pandemic has caused a total of 6 waves of global human infection, probably about 7 given the recent spike in China, and in the coming months, if the international community doesn't treat this accordingly. The largest wave between January 2022 and April 2022, about 30 million people became infected. And about 6 billion people have received at least one vaccine dose since the beginning of January 2021. However, throughout this extended time period, into 2022 of October, the SARS-CoV-2 genome has a time-independent constant mutation rate, going at about 3 nucleotide substitutions per month. That is, 3 nucleotide substitutions were fixed in the virus population every month, or about 29 substitutions per year. We denote this as a trend line in C, or the slope of the data, in which the mutation rate is the slope, that being the number of mutations that are observed per nucleotide site per month. We can ask ourselves a variety of questions from this data. So first, does the infected human population change the virus mutation rate at the genomic level at all? Does it just keep mutating like nothing's happening? Also, if the virus circulates in the human population for long enough, will substitution rate slow down as it becomes more acclimated to its host? At the beginning, the virus may mutate more rapidly in acclimating to a new environment, but once it settles down, it will start to slow down its mutation rate. The second question is, does the vaccination slow down the virus substitution rate? Or does it speed it up? Does it change the virus's behavior at all? Now, the answer to these questions is no. The virus seems to ignore these factors and mutate constantly over time. This is puzzling. What can explain the constant observed mutation rate for this virus? Is it due to neutral selection only? Under neutral selection only, which we'll get into more later, we saw earlier that the mutation rate is equal to the observed mutation rate, the substitution rate. But we must define the mutation rate first, and what is that? During transcription, RDRP can make mistakes. And these mistakes may not be readily picked up by the proofreader, the exoribonuclease that I also mentioned. Therefore, some mutations can be left behind over the course of the genome. RDRP can therefore make a random mutation at any nucleotide site with a putative constant mutation rate, that is mu, which has a probability to be fixed in the observed population. This is the substitution rate called C. Now, assuming neutral selection only, the observed substitution rate C is mostly due to neutral mutations. They don't do anything. While positive mutations can increase the substitution rate, they are incredibly rare and they don't really impact C that much. Deleterious mutations are removed, so they obviously do not make a contribution. Therefore, we would most likely see the mutation rate as a result of neutral mutations only. Let us imagine a scenario of how this neutral selection would look for this virus. Let's say that in a really, really small scale, we have 3 virus strains, NC, that each replicate with a chance to induce mutations in the population prior to neutral selection. Let's say each of these strains produce 3 progeny. Each of these 3 progeny bear a mutation that's in the red circle. So now we have 12 virus strains. The probability of mutation is about 3/12. The population, called NE, contains both mutant and non-mutant strains. Obviously, the parents that just copied themselves are not mutated. Now, under neutral selection from the host, that is a blue box, the host cell can induce an immune response to help get rid of these virus strains, or the host cell may die and kill the viruses with it. However, each mutation has an equal chance to be observed and fixed in the population after selection. This is called p-fix, which is about 1/4. And the sequences selected against do not end up in the population after selection, which is NC. The cycle then repeats after about 5 days. So we basically see that only 4 strains are left. We actually see that 2 of the non-mutant strains are killed off, and 2 mutant strains of each of the triplets also get killed off, leading a total virus population of 4. Now, from the population after selection, we can actually observe these mutations through genetic sequencing. And given that over time, the mutation begins to accumulate from all the genomes we sample, we can calculate the observed substitution rate. Assuming neutral selection, we would thus expect that C is equal to mu per unit time. The observed genomic mutation rate for SARS-CoV-2 in our data from December 2019 to July of 2021 is about 24 nucleotide mutations per site per year, or about 2 nucleotide substitution mutations per site per month. Again, it looks pretty linear. Now that we've gone over the 3 selection types, which are negative, neutral, and positive, and we've gone over what neutral selection looks like, let us focus on the 3 big theories of molecular evolution and see if any of them can explain the constant mutation rate of SARS-CoV-2. It should be noted that one side, believing in natural selection, fiercely butts heads with the other side, neutral selection theory, which sparked the selectionist-neutralist controversy 50 years going on. The first theory is selectionist theory, which was developed by Charles Darwin in 1859. Now, under selectionist theory, or ST theory, only the fittest species survive through adaptation and successfully reproduce. You probably know this familiar coined term. So in this case, you probably have learned that the giraffes with the longer neck who reached up to get the fruit survived and fared better than the giraffes with the shorter necks. It adapts to the environment and therefore can survive. Now, a species can only undergo negative or positive mutations under ST theory. Thus, most observed substitutions are positive and increase fitness, known as yellow. They arise through adaptation to the surrounding environment. However, as environmental variables change, like introduction of the vaccine, we would expect the SARS-CoV-2 mutation rate to be time-dependent and respond similarly to the environment. This is shown by the hypothetical data in blue coloring. However, clearly this is not the case, as its reported genomic mutation rate is time-independent. Therefore, it seems that SARS-CoV-2 does not follow the selectionist theory. We will revisit this later. Fundamentally different from selectionist theory is Kimura's neutral theory, KNT, which was developed by Mutsu Kimura in 1968. Under KNT, only the luckiest species survive through random chance and genetic drifting instead of adapting to the environment. The KNT sparked the beginning of the selectionist-neutralist controversy on which theory correctly described the molecular evolution of all species. We must note the change in the selection type distribution for the KNT compared to ST. We notice that the neutral selection in green makes up a significant chunk of the observed substitution mutations, whereas positive selection has become extremely rare. From what we have said earlier, we would expect a linear sequence variation line, and its slope is a constant mutation rate for this virus, which we observed. However, if the SARS-CoV-2 genomic mutation rate does not change over time, and does not change with vaccine exposure, then why must we keep developing the vaccine and the drugs? We therefore wouldn't be changing the behavior of this virus. This does not make sense because we still continue to develop vaccines in combating the more recent strains of COVID-19, SARS coronavirus 2. Information on why the KNT does not fit this virus will be explained shortly. A further adaptation of Kimura's KNT theory is the nearly neutral theory, which was developed by one of his students, Tomoka Oda, in 1992. ONNT states that most observed substitutions are nearly neutral and slightly deleterious. But what is the difference between slight deleterious and strong deleterious? Slight deleterious substitutions can actually be observed in the population with a time-dependent rate, which depends on the effective population size, i.e. the infected human cases, as shown in the hypothetical blue data. Strongly deleterious mutations are not observed. Therefore, they are not substitutions. The obvious reason why the ONNT is not followed is that, again, the observed SARS-CoV-2 mutation rate is time independent and does not depend on the virus population size. We arrive at a conundrum. We just went over the three big theories of molecular evolution, and it seems that none of them, save KNT yet, can totally explain the mutation rate of SARS-CoV-2. So then, what can explain it? It seems like the selectionist-neutralist controversy is not solved. Well, to figure this out, we should actually examine the individual subunits that make up the genome. That I mean the nucleotide level and the functional gene level. Do they exhibit the same selection pressures as a genome? Do they exhibit the same mutation rate as a genome? The answer for that is coming really soon. Okay, so if you notice in each of the three big theories we just discussed, each has a different selection type distribution, but none of them say exactly what percentage of each selection type would likely be observed in a given species. So we're actually going to figure that out. But how would you do that? So say you wanted to do this for each gene in a genome, or even the genome. Looking at SARS-CoV-2, we saw that it mutated about a very linear rate. So assuming neutral selection, we would predict that the observed substitution rate, C, is nearly equal to its mutation rate in the population, mu. By taking C over mu, it's approximately 1. We denote this as neutral selection. We do this because we want to establish a baseline. We want to know, are certain genes mutating faster or slower relative to the genome? To do that, we have to calculate the observed substitution rate of each gene, or whatever you want to do, even at the nucleotide site. And then take the ratio with the genomic mutation rate. So, if C over mu is less than 1, we say that this is mutating slower than the genome, and that it's under negative selection. However, it's also time dependent. If it's mutating about the same rate, C to mu would be approximately 1. It'd be under likely neutral selection. And this would be considered more time independent. If C over mu is greater than 1, we could say it's mutating faster and is positive selection, also time dependent. This method can be applied anywhere in the genome, even in the coding or non-coding gene regions. Now here comes the big hurrah. The near-neutral bound selection theory will help explain what we observe from the SARS-CoV-2 mutation data. From the NNBST, which is the acronym we're using, this likely means that each nucleotide site within the whole genome of SARS-CoV-2 did not exhibit the same mutation rate as we observed. To show this, we counted the total percent mutations, the observed mutations, within each site, all 29,903 nucleotide sites. Over 11,000 SARS-CoV-2 genomic sequences from infected human patients from the NextStrain database. This is combined over 3 sets, and we chose NextStrain because they basically handpick representative genomes that are complete and accurately reflect what are likely observed within the virus population. We then calculated the average percent mutations over all these sites, mu, calculated C over mu for each site, and generated a probability distribution for C over mu. Finally, we calculated the probability of each site experiencing any number of mutations, i.e. if sites exhibited 0, 1, 2 mutations, up to the total number of observed mutations. We then categorized them. So, to give you an idea, if a site did not exhibit any mutations, that means the C over U is 0. The more mutations you observe and categorize, C over U increases. Interestingly, an L-shaped probability distribution was observed for this genome, indicating that most of these sites did not exhibit any mutations. About 60% of these sites did not exhibit any of these mutations, and that the proportion of neutral, near neutral, that means weak negative and weak positive selection sites, as well as strong positive selection sites, those would be spikes in the data above, are incredibly rare. If we assumed that KNT was followed, what would this look like? Well, under KNT, each nucleotide site within the genome should be either strongly conserved, i.e. there is no mutation, or exhibit neutral substitutions, giving the following distribution below. If you notice, the approximate percent of mutations for each site, if observed, would be almost 10%. However, when you convert this to a probability distribution, it exhibits a Poisson distribution curve, indicated by the blue hypothetical data, indicating that C over mu would likely be around 1, and that increasing, decreasing mutation rates would become less and less probable. However, clearly, this virus does not follow KNT. Now, we're going to do what has not been done before. We are going to quantify the percentage of selection types within different parts of the genome, including the coding and the non-coding gene regions. We use C over mu to do this, and we wanted to check the amount of particularly weaker selection types. Why? Because although we already show that the Ota's nearly neutral theory was likely not correct, it did say that the amount of nearly neutral substitutions were mostly slightly deleterious. We wanted to see if that was the case. So, from this, we combined all the coding genes together, all the structural, accessory NSPs into all TR, or all translated region, as well as the all non-coding UTRs, all UTR, and TRSs, all TRS. Both weakly negative and weakly positive selection types were approximately present in each case. Thus, ONNT, ST, and KNT are likely not followed for this virus. And in the last little bit, we noted that the selection types for each coding gene were also calculated and are shown below. In particular, the 5' UTR, 3' UTR, M and ORF1ab gene regions are under negative selection. This means that if a site is under negative selection, these could indicate they are functionally important and that mutations that are changing in these regions could be very, very deleterious for this virus's life cycle. Interestingly, S, ORF3A, ORF8, and N are under positive selection. This may mean that they are likely responding to the vaccine response. To some, although we do see some elements of ST, KNT, and ONNT being followed for SARS-CoV-2, none of them are exclusively correct. The NNBST serves as a hybrid theory, which incorporates the fundamental aspects of each theory. From ST, successful reproduction comes from being able to adapt. From KNT, being successful comes from being the luckiest. These are both important concepts which can help explain the virus's evolution. But how do we explain the observed constant genomic mutation rate in the population? We saw earlier that at the nucleotide level, that we saw each site had a different mutation rate. But what's more helpful is looking at each gene, because those are actually more intuitive. Each gene gives some sort of functional importance. So by looking at the potential mutation rates of each gene, we might be able to solve this little conundrum we have. Now, when we applied our C over U method at the gene level, it turns out the ST theory seems to be observed. The observed mutation rates of each of these genes were time-dependent and rather lower or higher than the genomic mutation rate. When we combine these mutation rates together, they balance out to generate the time-independent genomic mutation rate. Thus, while KNT can partially explain the linearity of its genomic mutation rate, it's not 100% correct. This phenomenon can be explained using the NNBST. Okay, so the NNBST seems to butt out the three big theories of molecular evolution. But why does the NNBST really matter? What do we really care about it? What can we take away from it? Well, based on the different mutation rates we observe from the genomic mutation rate, we can actually go further, go into each gene or each protein, and see why it's mutating faster relative to the genome. See what regions are responsible. And in one really great case is the S protein. The S protein is basically the key to unlocking its way into host cells and infecting them. So basically the virus uses the spike protein and attaches to a cell surface receptor called HACE2, which is the lock on a host cell. Allowing entry, SARS-CoV-2 can infect the cell and initiate viral replication. Below is a solved structure model of the S protein, colored by domain. Domains are different regions of the protein which carry out different functions, which can explain its general function. So the key to the S protein is actually what's known as the receptor binding domain in green. It attaches to HACE2. Each of the other domains perform various different functions. To combat the virus, the vaccine is actually the RNA blueprints of the SARS-CoV-2 S protein, or spike protein, which is then wrapped in a lipid nanoparticle, which allows it to, which actually protects the vaccine from the extracellular environment, as well as the host cell environment. S protein is also a good antigen, so therefore it's actually quite good for developing antibodies against. When the vaccine gets into the cell, the host immune system will recognize the spike protein and develop antibodies to prevent future infections. But despite the high effectiveness of the first vaccine rollout in January of 2021, we've since needed to develop new versions of the vaccine to combat newer variants like Omicron. Why though? Is the S protein adapting to the vaccine and therefore that's why we keep updating it? To figure this out, we looked at the S protein mutation rate, the S gene mutation rate, and noticed that after the 10th month, that's when the vaccine started to roll out. We noticed that its mutation rate started to increase. This is likely an indicator that it's responding to the selection pressure to the vaccine. A selection pressure would likely prevent virus infection. It basically would prevent HACE binding to the spike protein, preventing it from getting into a cell, and then the extracellular environment destroys it. However, if it's adapting to the vaccine, antibodies generated by the host can't recognize the spike protein mutants, and it just enters as it always has. To figure this out, what about the S protein is actually doing this, we need to look at the individual domains, particularly the receptor binding domain. And we're going to use NNBST theory to prove it. By using NNBST for each protein, we can show that the vaccine can still save us. We can figure out why we have to keep developing new vaccines. So indeed, from the 11,000 genomes we sampled, we observed time-dependent substitution rates throughout each domain, different colors, in the S protein and at the amino acid level. The measure of mutational change was measured using KAKS, where Ka is the number of non-silenced amino acid substitutions which change protein sequence, and Ks is silenced amino acid substitutions. They do not change protein sequence. KAKS is actually a measure for assigning selection pressure to a protein or a given region within the protein. It kind of works similarly to C/U, except KAKS only works for coding genes, not non-coding. That's an advantage that CU has over KAKS, which in the paper we actually go into how we validate C/U in assigning selection pressure towards non-coding regions. Now, high substitution rates are designated as spikes in the colored spectrum above and are written explicitly in the protein model below. Now, notably, the receptor binding domain in green has the highest observed substitution rates out of all other domains, indicating they have increased mutation rates occurring in functionally important regions of the protein. These high substitution rates are likely in response to the vaccine exposure, as well as increasing adaptation to human populations of different genetic backgrounds, ages, and ethnicities. This is probably more towards earlier in the pandemic. By identifying mutation hotspots, we can develop better immunotherapies and small molecule drug treatments to help stop this pandemic. What's not as intuitive are regions that are highly more conserved, like transmembrane domain and TMD, as well as some regions of heptad repeat 1. These could also be considered as drug targets because if they have mutational change, maybe the virus cannot handle those changes and just be killed off through negative selection. Despite this, from all of this, NNBST has the power to sample time-dependent and time-independent features of the observed mutation rates at the nucleotide amino acid level, within each gene and in each coding and non-coding segments of the genome. In theory, this can be applied to any region. I just want to give a good acknowledgement to everyone involved in this project. On the left is Dr. Chan Wu, who's my PI advisor and fundamental in coming up with a theory. I helped brainstorm with him on some of the ideas, as well as help write a majority of the manuscript. From left to right is Mursalin, Mike, Drumibin, Philip, Emily, and Dylan, who helped us in the early stages of the project. Without them, none of this would be possible. As well as funding from the New Jersey Health Foundation, the National Science Foundation, and XSEED grants, as well as Anton II allocation. Again, without their support, none of this would be possible. Some final acknowledgements for metadata compilation and manual counting that were not listed in the picture are Lucas Bennett, Catherine Hausman, Annie Tran, Miraj Amin, Justin Carbone, Julia Gabriel, and Kenneth Bouilin. For early C-code compilation is Brian Chen, and additional insight and valuable comments, Dr. Yang Chen.